So welcome everybody. So today with us we have Professor Andreas Bender, who is working in a department of chemistry, but he's also very active in startups, assume related to chemistry and bioinformatics that he's an expert in. So he's Chief Technology and Informatics Officer at Pangea. I hope I pronounced that well.
No,
And advisor at a few other startups, mostly around drug discovery pipelines. Right. So we are really looking forward to hear Andreas' thoughts on how AI is being used in drug discovery. In particular, what we specialize in, active learning. I hope you'll be able to cover a bit of those methods. We know that they are starting to be more widely used in drug discovery. However, we are fairly ignorant about how exactly. So, yeah, looking forward to that.
yeah. Thanks for your intro. I will touch on active learning as well. The majority of the talk is actually about the domain because it's very important in drug discovery, life sciences data. It looks quite different, yeah, and that's actually a very important aspect. There will be a bit on active learning as well. Yeah. Okay, so yeah, this will be about AI in drug discovery. I'll talk about data, methods that are used, as well as pitfalls. Because if you read news articles, AlphaFold, and I mean, there's many articles right now, you actually would believe that AI in drug discovery is now the big breakthrough, yeah. But if you look at approval numbers, clinical approval, you actually see hardly any change. Okay, so obviously there's a disconnect, and I will discuss where that might come from, what we might be able to do, and so on. There's actually two articles. So as Fiore said, there's actually I'm involved in a couple of companies. So this is an academic talk. There's actually two articles we wrote a few years ago with Easy from the EBI about AI and drug discovery that touches on the data aspects in more detail. So in case you're interested in life science data, why does AI actually not work as well as an image recognition and so on? It's described in those articles in Drug Discovery Today 2021. Can really recommend to give those a read. It's written in a way that is really understandable by pharmacologists as well as computer scientists. It's explaining all the aspects of what is particular to the data we have. So that's the outline. So where are we? AI and drug discovery. What's the current state of that actually? How do we know that a method works? Validation is tricky in our area, and that is difficult because you never quite know how good your model is. I will explain why. It's different from other areas where images, for example, yeah, because you have this thing is called chemical space. It's undersampled. It's very high dimensional. You don't know underlying distributions. You don't even know what is out of distribution, what is not. Yeah, so the space we deal with is really, really tricky. I will explain what that means. I will talk about data, and that's a big shortcoming in AI and drug discovery, in my opinion. It's easy to build models. It's very difficult to build models that matter. I will tell you that, what I mean. And then talk a bit about generative models, active learning. How do big pharma companies actually use deep learning? Yeah, is it now implemented everywhere? Where do things stand? Then we'll talk about some aspects of psychology as well. I mean, if you of course, right? I mean, humans don't exist in a vacuum, they exist in society. And there's then quite a lot of drivers that are not necessarily scientific, and they can be even stronger than scientific drivers. So that's the areas I will cover. So where are we? So in the 80s, there was the first wave, I think, of computers and drug discovery. That's a Fortune cover, 1981, so 40 years ago, even more than 40 years ago now today. And that's where Silicon Graphics was founded. That was actually the first company that did proper computer graphics. Was also very impactful in visualizing protein structures, for example. I still remember some of those machines as well. In Cambridge, when I did my PhD, we had those. And here, the next industrial revolution is proclaimed, designing drugs by computer at Merck. So that was very structural, very visual. Doc, so one of the open source softwares where you can dock a ligand into a protein. Yeah, because that's how drugs work, right? You have a compound that inhibits, for example, an enzyme. Yeah, and that was written open source in that year. So that was very graphical, very structure driven, and very long ago. So that's my point. Yeah, people have analyzed computers, data using computers in drug discovery for at least 40 years. There's even 100-year-old articles that analyze relationships between structure and output. Yeah, of course manually, but yeah. So the whole field is quite old. So in the 80s, very structural, very graphical. Around 2000, the human genome sequence was published. That was much more bioinformatics, data-driven. Bioinformatics became a household name. There was the biotech boom, right? There was then the big crash in 2000, of course. 2000, very much data-driven. So in the 80s, graphical, structural, visualization. 2000, much more data-driven. And that's some of the headlines from today. It says here 2018 to 2020. I didn't update those because it's honestly, it still reads exactly the same. So how AI is changing drug discovery and so on. And you see, in some sense, the terminology is quite similar. What did change? I remember when I did my postdoc at Novartis, 2006, 7, Novartis was cleaning up their data. Yeah? So more data, ontologies, controlled vocabularies. Yeah? That all that was not around 1981. It also wasn't around 2000. That's around now. Yeah. So companies actually know what they know. Yeah. So you can use it for training models. The right models we need to see, but at least companies know what they know. Ontologies and so on. Yeah. That is actually something that really happened since then. So it's possible to generate much more data. You can, if you have an organism, you can now do what's called single cell sequencing. If you have embryo development, for example, right, you have fertilization of the egg, you can actually do single cell transcriptomics. So in some animals, you can track every single cell, how it develops, up to hundreds of cells. Yeah. So that's fascinating. So you can actually track embryo development on the single cell level. Yeah. Really fascinating. Also with cancers, what you can do is you have a cancer, you can analyze every single cell in the cancer and the environment. Yeah, so you have a tremendous amount of data. So you can actually characterize systems much better than you used to. The problem is drawing conclusions, actionable insights. Okay, so we sequence all those cells. Well, what next, right? You have very high dimensional data. You never have enough rows, and you always have too many columns, right? So not enough samples, and you have too many cells and too many individual genes. Yeah, so it's too high dimensional. You still have that problem, yeah, because getting enough rows, so samples into your system is very tricky. That's a very common problem. And to come to actionable conclusions. So what happens right now is you use data and knowledge, so data from single cell sequencing, as I just described, biological knowledge, yeah, to gain insights that are actionable. But that transition from data to actionable conclusions, that actionable insight, that is actually very, very tricky. Yeah. What is an early stage assay? So test system and the test tube I can run to extrapolate to humans later. So which type of cancer patient can I now treat, for example? Yeah. And that step, that is the tricky one, not generating data. I don't want to have terabytes of data. I want to have 1 bit of data. And that bit jumps to 1 when it's the right compound going to the right patient. Yeah? So it's not about more and more data. Yeah? It's about finding out which variables matter. And that is the tricky part because the whole system is so high dimensional. That it's very difficult to identify that. And also with computational methods, because you always undersample your system, you always have enough columns, too many columns, too many variables in your system. So what is my point here? AI and drug discovery is quite old. Headlines sound similar. We have more data, but to drill down to actionable insight is actually the tricky part. Yeah? And that is something people still fight with. What's the gene driving a disease? What's the target we want to tackle? Okay, to come to those very simple insights, yeah, it's very tricky to go from terabytes of data to that decision. It's that gene. Okay, that is a tremendously difficult step. Okay, and that is what matters because you want to find out what is the target you want to treat in a disease. Yeah, want to modulate in a disease. Okay, so we can do many things. We're good on quantity, but less good on drawing actionable conclusions from the data. That's very, very tricky right now. Yeah, Human Genome Project. Yeah, 2000. Some of you might have been in a conscious state already, depending on your age. But I remember that, and I was fascinated because we know now the building blocks of biology and so on. Yeah, of course, it turned out to be different because not about the building blocks, it's about interactions. The interactions in the cell are different depending on cell state. If you eat something different in the morning, your metabolism is different. Your cells express different genes. Okay, everything changes that system. It's not static, not at all. Okay, and that was underappreciated. So, but to come to the expectations, yeah, there was the expectations. We would have 3 or 10,000 drug targets. Humans have 23,000 genes, by the way, so that's the number of building blocks. But then people discovered there's different proteins that are formed from those genes, maybe 300,000 genes. And then imagine you have 300,000 by 300,000 proteins. They can interact, and you have a time element. Okay, so you get a point how high dimensional the whole thing is. But that was the expectation to have 3 or 10,000 drug targets. At that time, it was 483. But more recent estimates said the number of drug targets is not 3,000 or 10,000. Yeah, it's 667. Okay. And so it's very easy if you're on a boom of something to believe, wow, the world changes. Yeah. Yeah, maybe it does, but actually quite slowly sometimes. Yeah. And it didn't happen. The Human Genome Project was great to get a map. Okay. But it was not good to go to actionable insight, what to target in which disease, because the system is dynamic. Yeah. The map is static, the system is dynamic, basically. So that puts another dimension on the data we have, underappreciated, very difficult. But that what fascinates me. How do we go from technology, data potential to applications? Yeah. And I will come back to some of the companies we start in that area as well. So there's quite a lot of money going into AI and drug discovery, maybe 16 billion total. No one knows that. That's quite a lot of money. Developing a drug, people say, costs 2.6 billion or so. So you would expect 8 drugs you could develop for that money. If any big pharma company set up their AI team, it's going down. The cycle is going down a bit. At Novartis, some other companies actually closed some of the initiatives already. Anyway, there was quite a lot of focus on the area, of course. What did we get for that? So what you see here is on the left-hand side, what they call AI native companies. So companies that say we put AI first. So we let the computers decide, basically. On the right-hand side, big pharma, top 20. And if you do drug discovery, you have different stages. So once you're in discovery or preclinical, so that can be test tubes, animal experiments, then you go into clinical phases, 1, 2, 3. First one is safety. So you dose healthy volunteers and see if something goes wrong. If that goes right, then you go into phase 2, the first efficacy phase. So you dose into diseased patients, patients that have the disease you want to treat. And the phase 3 is like phase 2, just larger. It's a larger cohort for better statistics. Okay. And if you have AI native companies, you see that most projects, the vast majority, is actually in very early stages where you don't go into humans. Okay. So you have test tube experiment that work. You have no clue if that works in humans. That translation that's the big hurdle. Looks very different in pharma, right? So you have many more projects that proceeded to the clinic. And so what happened actually is that there's many companies that focus on where we have data. We have lots of data in very simple test systems. So we'll come back to that. Biochemical experiments. Yeah, it's very easy to measure. It's very easy to label. Yeah, supervised learning. You need to label your data. And so there's quite a lot of focus on preclinical data, very simplistic data. Yeah? In vivo data is complex. If you or any of you takes a drug, okay, how fast you metabolize that depends on, again, what you've eaten before in the last few hours, on your CYP, so cytochromes, certain enzymes that metabolize drugs, expression levels. Yeah? So on genetics, basically, it depends on which other drugs you take and how old you are. It changes based on time of the day and so on. Okay, so and all of those factors, they actually make it very difficult to handle in vivo data, as it's called, clinical data. But in vitro data, test tube data, you just put a compound and a protein to your system. You measure, does your compound block that enzyme? Yeah, you get a number, you can reproduce it, very easy to model, right? And there's quite a lot of focus on those companies, by those companies, on those very simple data, but they are not what matters if you go into the clinic. And that's what we call translation, to go from very simple test systems to go to the clinic, okay? And that's actually what many startup companies didn't do yet. It's happening slowly right now. There's in silico medicine, they now claim a phase 2 success. Helix, so the company we started, also goes into phase 2 now for neurofibromatosis. So it's a brain disease. But that's the proof point is still outstanding, okay? And I think AI and drug discovery has been around really for 10 years, let's say, or even now 14 years, depends on how you see that. And that's really the proof point, the clinical proof point. People get nervous. Yeah? People think, okay, we need to see that now. That there's really something changing. And that is not yet happening. If you look at approval numbers of drugs, that's not really happening yet. Um, Funnily enough, so you remember the headlines, right? 2018 or 2020, AI was changing drug discovery. 5 years later, there's some headlines from Nature Medicine article. There's no shortcuts. Trials are in the early days. It's all very difficult. Okay, so people really roll back a bit and say, "Hey, okay, it doesn't quite work as well as we thought." But of course, you need to be optimistic, right? New technology, you need to be optimistic in the beginning, and well, do the best and hope the best, right? But the clinic is tricky. And drug discovery, okay, it's not about models. It's about what does it tell me about the patient, okay? So you need to understand your application area. You need to understand what matters for your final decision, for your final purpose, for final use case, okay? And it's not the models. The models are means to an end. It's the final use case is always the clinic. Yeah, conclusions are lots of activity in early stage pipeline, lots of money. The question is, where's the novelty? So there's out of distribution prediction is difficult, right? So if you look at the chemistry of what those AI first companies do, it's very similar chemistry. So to what was done before. Extrapolation is difficult out of distribution prediction. Data is a limiting factor. And there's also quite a bias by media. So there's always whatever, even New York Times headlines, right? The first AI designed drug and all that. In drug design or drug discovery, you have a 10-year process, and there's 1,000 humans involved in making decisions. And if your model predicts something, right? There's humans shortlisting and the synthetic chemists deciding that compound we cannot make, we need to make that one or make an analog. There's human interaction, right? So you can never really decide or come up with a conclusion that the algorithm, right, designed the drug. Even that term doesn't make any sense, because that would imply computer makes a prediction, and that's what you carry through. It never happens. You always need to optimize your compound afterwards and so on. So the application of algorithms in drug discovery looks different in this particular area compared to many others. Now, validation. So you need to validate models, right? So you have your whatever training, validation set, test set split, and so on. We can also, in principle, do that with the data we have, but there's real problems with doing that. What are those problems? The labels are either what we call in vitro relevant. I will come back to that with the visualization. So they relate to this test tube data, yeah, unconditional data that is simply putting a compound on a protein, for example. We get a number, reproducible, but it doesn't consider all the factors that matter for that compound in a human, because then you don't only have activity against the target, you have uptake in the stomach, solubility, bioavailability, right? So and that is a very conditional system. So we basically have a pick your devil situation. Either we take in vitro labels that don't extrapolate, but we can label data, build a supervised model. And if we have in vivo data, it's very conditional. Yeah, they depend on genotype, on the dose, and so on, right? So, and none of the data is what really matters for us, right? One is proxy data, but it's not what really matters. The other one is really conditional. Depends on, I don't know, 20, 50, 100 or whatever conditions, yeah, of the genomics of the patient, how highly you dose, and so on. Okay, so we cannot label our data. We cannot label the data that matters properly. Yeah? And if you cannot label, you cannot measure how good your model is. Validation is costly. So retrospective validation basically is not meaningful. So you need to prospectively validate, use that in models. But that's actually very costly. You can never run a case control experiment where you do the same project with the AI method and without. Yeah, that costs you 10 million. No company would do that. So there's still prospective validation. And there's a particular problem in chemistry. It's difficult to sample chemical space well. I'll illustrate that on the next slide. I'll show that on the next slide.
Sorry, hey, can you just clarify what you mean with prospective data
Mm.
exactly?
Yeah. So if you so you have in a pharma company, you have 100 projects you run per year or so. That is your historical data. And prospective means we have the new. So tomorrow we want to work on depression on a particular protein target, and that is our new project where we have little data. We don't know how well our models work, but this is the new case we work on. Yeah. So retrospective validation, we do some training validation test set splits based on historical data. Prospective is really a new case where the model is required to make out of distribution predictions effectively.
Right,
It's new chemistry. It's a new ligand, yeah, new chemistry, new target we work on. So it's a new project. And that's, of course, in pharma what real world projects look like, right? We don't work on what we worked on 3 years ago and predict for that project. We predict for new chemistry. Yeah, so we require out of distribution predictions that don't work very well. And even to generate then all that data experimentally to see how good your models were is very costly. Yeah, because you would need to, it's easy to make predictions, but you need to make the compounds, you need to run the experiment. You would also need to, in principle, put the compounds into the clinic, right? Because that's what really matters. Yeah, but that then gets very, very costly. You cannot do that with hundreds of compounds. So you don't have enough data to validate on new projects how good your model is, basically.
Okay. So essentially test data potentially out of distribution.
Yeah, but it's yes, but it's a test. So we have historical test data, but the thing is, chemical space. Actually, I think I might go to this slide. So the point is, yeah, actually, I just put this one on. So this is what chemical space is very high dimensional and undersample. and behaves very differently in every area. You have what's called in chemistry, it's a scaffold, so it's your core of the molecule, and you have variations at the edges. Yeah? And quite often, your core determines how variations around the edges of the molecule, what the impact of that is. Okay, so which means in every galaxy here, which is what we would call chemical series, so it's a set of compounds that are similar and have only slight variations. Within that set, you can actually predict variations. But if you don't have any data to actually characterize the underlying properties of that series, you cannot extrapolate to it. Okay, so it's very easy to extrapolate locally, so slightly out of distribution, if you wish. Yeah? So that somehow works, but to jump to a completely new area of chemical space, which is what you would do in a novel project, yeah? That basically doesn't work. Okay, so and that is the tricky part because that space is 10 to the 60 or so molecules. We can buy 10 to the 10. We have data sets 10 to the 6, but 10 to the 6 out of 10 to the 60. And so for most galaxies series, you don't have a single data point, okay? And that extrapolation doesn't work. And that means that your performance is also not very meaningful because you train on this set, validation set. Your external test set is often not external because it's drawn from the same underlying distribution. Yeah. So there's still some similarity. It's not what the next project is. The next project would be here, very different chemistry, really out of distribution. Your external, so it's not external, that's the point, test set, yeah, is actually very close related because quite often you just do splits of data. In chemical space, it doesn't work. In chemical space, that does not work because your novel data points are completely different chemistry, which you cannot extrapolate to. And that is different from other areas where you actually sample your space much better. It's the size and this jumping to a new galaxy difficulty, basically, that makes modeling in chemical space very difficult.
So just a follow-up question. Why is it a different space? Is it because perhaps, I don't know, different tissue is targeted, where you want to try out new kind of compounds to treat something?
Yeah, so if you have this space means here it's novel chemistry. It's a novel scaffold, as you would call. So this core of the molecule is novel. You need that for many reasons. You want to target a novel protein, which requires new chemistry. You want new chemistry because of IP. So you need to commercially, so to speak, have novel chemistry, or your properties of your molecule. It binds to the target, but it doesn't get absorbed, maybe. Yeah. So you need to do something with your molecule. So you need to vary your molecule, yeah, in order to have an optimal profile of 100 or so properties of being active on your having the effect that you want, being absorbed and so on, as well as being non-toxic. Yeah? And then you need to make variations, which means you need to vary your chemistry. Otherwise, you don't reach your goal, your target product profile, as you would call it. No, but feel free to ask. I mean, anyone else as well. Yeah, but that's very characteristic for chemical space. So that extrapolation basically only works in some cases, only works in very, very few cases, basically. Sometimes you have descriptors that capture underlying relationships. In many areas, we don't have them. Yeah. So in some cases, you have what's called physical chemical descriptors, size, polarity, weight, and so on, that actually describe the underlying properties well. Then you can, to an extent, extrapolate. In some other areas, we don't have that. So basically, we have no clue how molecules behave in different areas of that space, which is large and unsampled.
Right. So I mean that's I mean physics, underlying physics should be kind of same and known more or less. So you cannot kind of build your models such that they have kind of good understanding of the underlying physics and extrapolate based on that.
It works in the areas where you have what's called solubility, let's say, or log D as it's called. So it's like where you don't have directional interactions, for example. In some areas you can, and that is actually one of the big hopes right now, physics-based modeling. Yeah. The problem is if you have an interaction of a drug with a receptor, it changes conformation slightly, then you have downstream signaling, and you have many molecules that interact. You don't know which ones, yeah, you don't know what's around the cell, what's the concentration, yeah. You cannot characterize your system properly as input for that physical modeling. And the physical modeling is actually very complex to do because you have large systems, millions of atoms, yeah, and that can go wrong quite easily if you don't parameterize properly. In principle, you're right. In practice, that's quite difficult. Yeah? It works in simple cases, but not in the majority of cases. Yeah. Yeah. Yeah, and there's also quite a disconnect. I've seen that. So I was in academia, but also we started companies. So that's what academia cares about quite often. You have a data model, predict accuracy goes up or F1 score or whatever. You publish a paper or you're happy your model is better. But actually, what matters in drug discovery is actually more the embedding, right? So you build a model based on, let's say, a bioactivity cutoff. You have a binary model, and you say at a certain concentration, my compound is toxic. Okay, you build your binary classifier, done, you're happy. But in a project, of course, things depend on what's the dose in men. If you have a toxicity classifier at a given threshold, and you dose much higher or lower in humans, the threshold doesn't matter. Yeah, so you would in principle need to retrain your model, but that's quite a process that in practice usually doesn't get done. So there's a disconnect, right, between the project context as well as the model that you build. You don't know the future use cases of your model. In other words, yeah, that's one way to characterize that. And there's also, on the other hand, there's a disconnect. You can predict something, and the question is, if you spend 10 million on a project, and then the computer says 80% likelihood your compound is toxic, you don't scrap the project, right? You spend 10 million. So the question is, what's your follow-up assay? What do you do afterwards? Okay, so that is then the important part. So what is the experimental follow-up? That's so I was at AstraZeneca at some stage running the computational safety group. That's even more important than having a good model. You need a good model, of course, but whether you have a slight increase in model performance or you have a better embedded pipeline with the follow-up assays, the latter is in practice what makes the difference. Yeah, that you can test tomorrow and cheaply whether the prediction is correct. Okay, so you can prioritize with the model, but then you need to follow up. And that's very important. Yeah, that embedding into a pipeline, into the input project context, as well as the follow-up. And there's quite often a disconnect in practice. And academia sometimes doesn't help either because it's only focusing on this box. Yeah, and then there's the question, well, what do you do left and right, basically? And how do you actually include novel project data? There's lots of engineering questions actually associated with that. Yeah. Yeah, let's skip that one. So Achilles heel, so it's data, and I alluded to that before. So, but let's come to that in a more structured way. So what we modeled here is, so that's the drug discovery pipeline. So you find a hit, as it's called, a ligand active on a target, optimize that. You do animal studies. You have this clinical phase 1 for safety, then for efficacy, larger efficacy study and approval. And what we modeled is change of time. So we are faster 20%. We have 20% lower failure rates, and we're 20% cheaper. And what you see here, and so we map time as well as cost savings on one axis, so we can visualize it like that. And here you see net savings, so higher is better. And what we see is better compounds, so higher quality of compounds in phase 2 makes a big difference. Okay, and that is unfortunately exactly not the area where we have our main data, which is here. Because simple test tube data, as I said, right? Easy to generate, easy to build supervised models on, but that's not what matters for the clinic. Okay, so there's a disconnect between where we have data and where we can label it and happily build models. And what really matters is clinical data, which is messy. Okay, if you have a clinical trial, whether even you have an endpoint definition, as it's called. So you put a drug into a patient, and then you say you have a primary endpoint, tumor shrinks, for example, right? Or tumor shrinks more than standard of care. Yeah. And whether your trial is successful depends on that endpoint definition. Yeah. So you cannot even say a drug works or doesn't work because it depends on the comparison that you subjectively choose. Okay. And so you cannot label that data, is my point. Yeah? You can dose differently. Yeah. You can put the compound to different patients. So very difficult to label data to train models in that area. But that is what matters. Clinical phases. Okay. So here it's easy to build models, but that clinical translation isn't there. Here you could get the impact, but you cannot really, it's very difficult to build models because you cannot label your data and you need labeled data for decision making. Of course, you can have unsupervised and whatever methods, but in the end, you need labels for decision making. You always need some labels. What do I want? What do I not want? Yeah, and let's see. So there's two types of data. So one is what we call in vitro test tube. The other one is in vivo. And that is a bit underappreciated sometimes, just to illustrate that here. So this is, I did my postdoc at Novartis ages ago. And so until then, I also believed labels, yeah, in the life sciences area. Compound is toxic. Doesn't make any sense because it depends on dose. The compound binds to a target. Well, maybe if it reaches the target tissue, Does it get into the brain to bind to the target? If not, completely irrelevant if it does that in a test tube. Compound treats the disease. Well, depends on dose, depends on the what's called disease endotype. In cancer, for example, right, you can sequence cancers, you have different genomic profiles, right? So does it treat the disease? Well, depends on the disease subtype, okay? So none of those labels actually make sense, and that's one of the problems in our area. And here, lots of text, you don't need to read through that, but the point is, if it goes to life science data, you have that on every level. In what pharmacology, a compound is not active on a target. You can have a myriad different types of effects, yeah? Activate the target, inactivate it, and so on. And often that's not characterized. So again, we cannot label. Disease as well, phenotyping. Quite often, we are still stuck in labeling diseases the way people did 100 years ago. It's very organ-based, yeah, liver cancer, for example, yeah. But that's not the biological mechanism. That's meaningless for classification. Yeah, we need to understand disease. Often we don't know. Yeah? We need to understand disease to be able to treat it and to classify diseases and label diseases properly. It's very often phenotypic, as we call it, observational, yeah, cancer of the liver, yeah, and not mechanistic, and so on. So computer science, very powerful, but is our data? Can we label? Drug treats the disease? Well, it depends, right, on endotype, which you often don't know. Like ligand is active against the target. Well, if it reaches the brain, it's very conditional, the data we have. Yeah? And that's very difficult to include in models. Of course, you can include that as variables, but in the end, you then end up with one data point per model, basically, because you have so many conditions, that you have very small subgroups. It doesn't work in practice either. So yeah, that's one of the problems. Yeah, so sometimes if you read machine learning journals, you see classifiers that approach questions then in too naive a way. Does a drug cause an adverse drug reaction? Yeah? And then there's a binary classifier, 89% accurate, whatever. But in practice, that is what your data looks like. If you have a patient with a genotype, certain disease endotype, who takes a high dose or low dose of a drug, then we might see an adverse drug reaction, which is an arbitrary ontology, by the way, that is very phenotypic, so also not satisfactory. Then we see it in a fraction of cases. 30% of cases have an adverse drug reaction. Some more severe, some less severe. Some, if you take another drug among old patients, right? People above 60 take on average 6 different medications. So maybe it's a drug-drug interaction. Maybe you see it in males, maybe in females. You have to, maybe after 3 months of dosing and so on. And so that is what real data looks like, right? So even water can be toxic. If you drink 3 liters of water in half an hour, you dilute your electrolytes, you have 50% chance of dying, okay? So, but would you in a machine learning model label water as toxic? Probably not, right? So just to illustrate the problem with data, and that's the main problem we have in the life sciences, actually. Yeah. So there's lots of disconnects here, and they are due to the data that we have, right, as well as lack of embedding of the model into the process. And in the end, it's always about the process. You don't just want to improve a model; you need to improve a process. So the question is, which data do I need in a model? What is the performance measure that matters, and what are the embeddings, right? And that's what you need to think about from the onset. If you build a model, it's not about the model. Or I speak now from the life science domain, but in that area, it's not about the model, it's about that whole pipeline that you need to think about. And there's also, so this is an illustration of why in my opinion, why AI in the drug discovery area didn't deliver in the way it did, as it did in other domains. So in the image domain, you have an object, you have a matrix, right? 24 bits or whatever bit depth, or here it's 1 bit, but you have a matrix basically where you can learn your convolutional network. So you have your edge detectors or convolutional layers and so on, and they operate directly on the matrix. So you're going to represent your image as a matrix. CNN operates on the matrix, and your object label is largely unconditional. So there's a cat or there's no cat. Of course, it might be partially hidden and so on, but it's still less conditionality than other area. So good connection basically between the object, representation, model, and label. Very different in chemistry and biology. So your molecule can rotate, it can react, can be protonated, and so on. You don't know how to represent it, yeah? So that is trial and error. You don't know how to model it, so you can have some feature space and then apply whatever random forest or whatever you want, and then your labels are conditional, okay? So you don't have that good connections, right? So you don't have this object representation as a matrix, CNN operates on matrix and so on. You have disconnects everywhere. Yeah? And biology is the same. So basically, in this particular case, also in speech, you can label, right? You have a waveform and you have a label, which word does that mean? In the other areas, you don't have that, right? So, and that's basically one visualization why AI in life science is much more difficult than other domains. And there's so that's one figure as well from that article I mentioned before. So there was quite a lot of focus on methods recently, or end-to-end learning and all that in the life sciences. But as maybe I made clear, is we need to actually go deeper, and deeper here means to see which data do we have and what's the question we want to answer. And for example, the question is we want to classify patients that have a certain disease. So we need to understand which gene is up or down regulated, for example. Then we need to generate that data. So not just data and then try to find a signal later, but the right data that addresses that question. And then we need to represent it in a suitable way, let's say a biological pathway or so. And then we have the method on top. Right? So there's quite a lot of focus here, but I mean, you cannot rescue up here what was messed up before already, right? So you need to go down to the bottom of that pyramid and then understand the disease, and then identify the data you need and so on, and go up again. It's quite often not done right now. There's quite a lot of dabbling around with the data sets we already have, not the right data. So I was talking to some people from, I don't know, Wellcome Trust and so on as well. I think what we need to do is we need to understand diseases to see which diseases are just at the level which we understand well enough to identify markers. Yeah. And those diseases can then actually be used in those areas. AI can then be used in order to identify a signal that characterizes that disease. Yeah? We need to have enough understanding, basically, that data makes sense. Just data will not solve our problems, yeah? But we need to have enough understanding so we can identify the right signal in the data using the right method. Yeah, explainable AI is quite a tricky area in chemical space as well, because of biases in the data sets. And that is painful to the extreme in the chemical case. I still remember that. So that's work I did in my PhD. Bitter compounds. Yeah. So compounds you eat and they taste bitter. And so we had a data set with Unilever, consumer goods company, of a few hundred bitter compounds. And I appreciate most of you are probably not chemists, but if you look at those molecules here, we see quite a lot of those rings here, 6-membered rings with OHs. They're sugar rings. Okay. If you ask the data or the model which features are characteristic for bitter compounds, you end up exactly with that, with sugar rings, because you see them quite open. Here's the open form, it's still a sugar ring. There's also sugar or open form sugar, not a sugar ring, of course. And because why is that actually? It's a confounding factor. Natural products are often bitter. Natural products are often glycosylated, so they have sugar rings attached. Okay, it's a confounding factor. It's a huge confounding factor. And so to do explainable AI in this space, basically, I would even say it does not work because you don't sample your space well enough. You have huge biases in your data sets, yeah, so you don't really get to explainable AI. Not a fault of any algorithm, it's a fault of the data sets. It's a fault of the data sets. Yeah. You have to be really careful with it. Yeah, and so we did start some companies as well. So Helix, I still remember when Tim, so you see on the picture, CEO of Helix, is now 70 people or so, compounds going into phase 2. When he came into my office, he suggested, hey, Andreas, let's do data for repurposing and so on. And I thought in 2014, a great idea. I mean, let's do that. But isn't it too late? Yeah, it looks 10 years later. In hindsight, it looks quite funny because of course, I mean, we know now well. I mean, things are happening now, so much later. So it was quite early. What's the learning? I think if you're in a particular area, you're quite on the forefront, right? You drive the whole area, but the world moves very, very slowly. Okay, so don't think you're late. And quite likely, if you're an expert in that area, you're in the first wave. Yeah, in the whatever, even before early adopter, whatever it's called, the slice before. Yeah, so the people who drive that field, right? So don't think you're late. Yeah, the world moves slowly. You need to come at the right time. Yeah, and that was certainly still early enough. Now Helix got funding and has compounds in the clinic. The other one is Farm Enable. This more fee for service. So we have computational algorithms that predict which chemistry can be synthesized and which targets they target. Yeah, very different business models. So Farm Enable worked with Heptarius and so on. So some companies in Cambridge. What we do in Pangea Bio, so we renamed recently, is we use compounds from plants with historical use. So the problem, as you might remember from the plot, is phase 2 in the clinic. Does the compound work when you go into humans? But plants have been used for as drugs for centuries, for millennia actually. And so what we do is we have a big database, knowledge graph actually, that contains plants with historical use information, the compounds they contain, the targets and diseases that are linked to those targets. So we can use historical use evidence, phenotypic evidence, plant acts on depression, for example, as well as modern medicine evidence, compound target disease links, in order to zoom in on the right compounds to select. And that is basically what we hope increases chances of success in the clinical phases. Yeah? Because there's evidence already that the plant has been used in 3 different cultures to treat depression. So there's evidence of phenotypic efficacy. And I can really recommend I mean, you're working in companies anyway, but to actually do research, but then also to translate that, because otherwise it can be a bit of an artificial system, like you've seen those flowcharts before as well. Few computational aspects, yeah, generative models, active learning, deep learning, and so on. So this is how people often right now see how drug discovery is being done. Design, make, test, analyze cycles. Yeah? You design a compound, you make it, so synthesize it, you test it in a test tube or some other system, but in an experimental system. You analyze the results, and then you enter design again. And the design is partially now done using generative models. So where you can have different types of generative models that now generate new molecules that are scientifically, chemically plausible, evaluate them using, for example, those docking algorithms, putting a ligand into binding pocket and optimize it and so on. Yeah? So you do this one many times before you make a compound in experiment. And yeah, that's where we were working on, Morgan Thomas in particular. One of the shortcomings of the areas is, like I mentioned before, prospective validation. There's lots of non-goal directed measures. For example, here, fraction of valid and unique and novel molecules, but that's actually not really what matters. Yeah, what matters, I mean, that also matters because you need to have a valid molecule. Yeah, unique. Okay, so you need to have explore chemical space, right? If it's always the same, it doesn't make sense. But the thing is, does it fulfill the objective, binding to the target? Okay, and in many benchmarks, that is actually insufficiently considered. Yeah? So there's certainly a shortcoming of proper validation in certain areas. So what we did is prospective validation experiments with Heptares, the company that actually funded Morgan as well. So we synthesized compounds, tested them to really prospectively validate. That's the only way applied in practice. Otherwise, you don't know if it's really working in a real world situation. And you can then see, do you actually find novel molecules, novel chemistry, and so on, scaffolds, as we call it. So scaffolds are those galaxies, like in the previous picture. You actually explore many galaxies and so on. But synthesis experiment, that's actually what matters in practice. Iterative screening. So we mentioned those experimental assays, right? So you test compounds in a test tube. In practice, it's more plates, 96 well plates or 384 well plates. So you can test many molecules in parallel. And some of the assays are actually quite costly to run. So some cheap assays cost you 20 cents per well, so 20 cents per test. Is actually one of those plates. So per well, so you put one compound in one well, costs you 20 cents or so per test. But some more expensive ones can cost you a few dollars. So you cannot test 1 million molecules. And then you do iterative screening with active learning. And you then have criteria like exploring areas of particular uncertainty, or so you can explore or exploit, right? And first you often explore, then you exploit later, in order to zoom in on the clusters, on the global optima, basically, of the most active compounds. And that seems to work. So what we analyzed here is over different iterations, where actually quite few compounds were selected. So you would expect by chance maybe such a line here, because it's very small sets that were selected in every step. You see that out of the highly active scaffolds, so 100% is all galaxies were identified, basically. Even in the beginning, you actually retrieve, with certain parameter settings, you retrieve quite a lot of the active galaxies. So active learning seems to make sense, and you have different criteria how to expand your into novel chemical space, and that's standard, I would even say, among big pharma companies right now as well. Implementations vary, but due to the cost of assays, That's actually anti-integration, better integration of computational methods and robots. Robotics is very important because you need to get hold of the samples, right? Until 10 years ago, robots were very slow. So you could do nice things in the computer, but then you need to collect 1,000 compounds to test in the next iteration. That used to be not possible, but that's now possible. So you can do that in a few hours in order to actually have active learning cycles also that integrate computational methods and robotics. Yeah? You need that part as well, that experimental part. Yeah, deep learning, so it can work well, sometimes works well numerically, but still doesn't address the underlying question, and sometimes also pushed in a biased way in publications. So here people did a target prediction. So you have tens of millions of data points from large bioactivity databases that link chemistry to activity against the target. So you have 1,000 or so compounds linked to a certain protein target, and you have on the order of thousands of classes. The problem is some of these sets are quite small, 10 compounds. Sometimes you have 10,000 compounds. Sometimes they are diverse, sometimes they are not. Very difficult to normalize between the classes. And that's a neural network. So feed forward, that's what it stands for, a neural network, but a deep network. And that in this case, in this particular study here, this study outperformed other methods. In practice, so when we worked in pharma companies and so on, quite often you have data from a project every week. You want to incorporate that into your model. And quite often, if you have deep networks in practice, that cycle, that update cycle, and so on, does not scale very favorably. Okay, so in practice, actually, I think random forest models quite often give you 90%, yeah, or even 100% or not of what deep learning models would give you in that area. But it's much easier to run computationally. So there's still a huge space for, let's say, random forest models in many areas of pharma, yeah, because you have those update cycles and so on. Yeah. And sometimes you have to be careful with those labels, what matters in practice. Yeah. Synergy between drugs. So we worked on that as well. Drug A and drug B, the effect is more than the what do you expect due to addition, let's say. It depends on what your baseline is, but let's assume it's addition, additive effects. Yeah. And that synergy, some people say, is good to have because you need lower doses of drugs, fewer side effects, and so on. And so we also predicted synergies of drugs. And so I had a deep network. It wasn't that deep, to be honest, and explored some other methods and so on, and predicted synergy scores. And when we published that, I thought there's actually a really good model. So we had a certain true positive rate, true negative rate, and so on. So what does that mean in practice? So 1 out of 2 positive predictions is correct. Well, 19 out of 20 non-synergistic predictions are also correct, can be discarded. Yeah, sounds quite good. Okay. But in practice, that is data that is generated on cells, okay, on individual cells. If you take a compound, you have different absorption in the body, you have a different concentration at the tumor, for example, right? So does all that matter? It's a very artificial system, yeah? So when I step back, and it's good in science to be self-critical, right? If you step back, does that translate to the in vivo situation? Actually, much less so, right? So you have to be careful what you model. There's a saying, it's more important to do the right thing than to do things right. Okay, so what do you do? What do you want to do? Yeah, and then to figure out the way to get there. There's no point in doing the wrong thing in the right way, right? You need to do the right thing and then figure out how to get there. Okay, yeah, and sometimes in publications there's my numbers are higher than yours, game. And there's also quite a lot of bias reporting. So that's, for example, an abstract deep learning with electronic health records and how much better things work. Yeah? But actually, if you look into if you compare deep learning to a logistic regression baseline, you see that the confidence intervals actually overlap. Yeah? So you have different OROC values for the different methods that you used. And if you compare those values, you actually see that those values are not fundamentally different, okay, between deep learning as well as logistic regression. So there's a certain bias in publication and how things are presented as well that I think is not very helpful in many cases to advance a method. Okay? Yeah, 2 final slides on psychology, and then I'm done here. So the bigger picture, of course, AI is where it is also because of human psychology. There's quite a lot of fear of missing out, also in pharma. The others also do it. Beliefs often drive decisions. Maybe they have the secret sauce, a bit tongue-in-cheek, but that seems to be then the ideal startup strategy equation. If you generate enough hot air but not tell anyone how you do things, And then you have fear of missing out from big pharma. That creates then the perception of the secret sauce, right? Because then everyone thinks you do something fancy, they don't know how to do it, right? And that is, of course, a bit tongue in cheek, but that seems to be the strategy of some of the companies out there. I think there's no real secret sauce. The secret sauce is good execution, actually. Not having the idea, good execution, that's actually what it is. And multiple levels, so individual psychology as well as organizational psychology matter. There's also quite a lot, especially in big companies, different effects. Everyone needs a winner, for example, right? After someone gave you 20 million to implement the AI strategy, your boss asks you, so what worked? It's unlikely you say nothing, right? Because everyone needs a winner. So, and then there's this overrepresentation of good results. It's a bias, right? So how do you know what works? But selective reporting of successes, of course, leads to everyone declaring victory, but actually no one knows what's going on. Difficult to advance a field if you cannot really compare. And you cannot compare because we don't have the labeled data and so on, as explained before. So we need to analyze our data, absolutely. AI is a valuable tool in the toolbox. But the game changer comes from the translational aspect, yeah? The clinic. So not modeling things that are easy to model, preclinical data, as we call it. The question is to address the elephant in the room, what really matters for better translation to clinical data. And we probably need consortia for data generation and sharing and so on. And the methods need to translate into reality. So it's not about the model, it's about the process, right? The embedding of the model into the process. Okay, I think that's all. Okay, thank you for listening.
All right. Well, that was interesting. I think I didn't e- expect that mostly it lies in data.
It does. It does. Honestly, I can tell you that's where it is. A random forest deep learning. Okay, fine. But the question is, what's your endpoint? Do you have what's the coverage
Yeah.
of your data? Yeah.
And it doesn't seem like it's in a way solvable because again, getting those final endpoint like in vivo data, it's extremely difficult, right? So do you see that happening like that's going to change amount of that data so that you can actually then perhaps at that point you could think of having machine learning models that would have broader set of inputs that you were saying like, okay, there's lots of environmental factors that are not considered at the moment, and then also the end metrics that you care about, translation in vivo.
Yeah. Slowly, very slowly, in the sense of better. So until 10 years ago, there was no clinicaltrials.gov, for example. That's a website that archives results from clinical studies. Okay. So at least, for example, that website exists. It doesn't have all the annotations we need for machine learning. Okay. At least that website exists. And the majority of studies is not published there, okay? That needs to be addressed. So more data, better ontologies, controlled vocabularies, okay? Then it could be used for machine learning. Then biological characterization of patients and diseases, yeah, that would need to be added. Let's say sequencing of every patient in the study and so on. Yeah? So the path there is in principle possible, in practice quite difficult because you need to do all the things which I just mentioned, and you can probably only do that if you really know which data to generate in a particular disease. And sequencing is not all. Yeah, it's maybe how do you actually do you sequence only the tumor or the surrounding tissue, for example? Yeah, maybe you need to generate other types of data and so on. So there's in principle the path I think is clear. In practice, it's very, very difficult, I think, to get there because there's both organizational barriers as well as scientific barriers right now. I think in areas where we can basic biomarkers, for example, they change clinical success rates threefold from 3 to 8%. Huge difference, huge difference. So where we can basically have a genetic marker that tells us that's the right patient to treat. Yeah? So we need to have that understanding to identify the marker. And then we, in principle, are already one step closer because we can characterize the biological system. Yeah. Then we know, marker up, that's the patient we should include. Yeah. But basically, it's a multitude of factors. Some are organizational, some are biological and medical insight to generate the right data. It is a long path. Honestly, it's a long path, a very long path.
Right. And those AI native companies, I mean, 10 years is a long, let's say, financial runway that you would need to keep at to actually yeah, I mean, to wait for that long to actually start having success. So is there already a lot of failures of these AI native companies?
Yeah, there's, I mean, until 3 years ago, let's say, it was quite easy to get money, right, for a nice story and nice team and so on. So that changed in the last years. Some companies failed, some companies emerged. I mean, Recursion bought Exscientia, for example, right? And there's some Exscientia was basically the poster child company in the UK in the AI and drug discovery company area. So there are some mergers, failures. I think over the next year we will see quite some failures because of how difficult it was in the last 2 years to get new funding rounds in. So I think there will be quite a lot happening. Yes, some failures we've seen. I think many are still to come. I think so.
Yeah. Okay. And is there kind of an updated view on that initial graph that you showed, like 2021? At that point, there was almost no drugs from these AI native companies that are in phase 3. Has that changed at all?
Phase 3, not so. There's this one combined phase 2, phase 3 trial by Recursion, for example. I don't think there was any readout yet. In silico put or claimed now success in phase 2A. Things are slowly moving forward, but I don't think there's a real proof point on a bigger scale. And honestly, I think because you need a certain sample size, yeah, to I think the next 5 years we still probably need to see more output. I think in 5 years the world will look different in that area. I really think so because of financial and clinical success reasons. Let's see. I think there will be things crashing, yeah, but certain things will survive, yeah. And also from the 80s, structure-based modeling that was invented in the 80s is a staple in drug discovery today, yeah. So certain things will survive. Also, having data organized, annotated, that's a plus as well, yeah. But it's not the panacea, yeah, for drug discovery AI. I don't think so.
Right. Anyone from the room or online?
We have questions, but unfortunately, we have to leave the room in 1 minute.
Yeah, we have to now.
Maybe we'll keep some of those for if we can get one-to-ones.
Yeah, sure.
Yeah.
Thanks a lot for this very sober view on artificial intelligence.
Yeah.
It echoes quite well what we are seeing on our side. I think there's also a bit of fear of missing out from the craze in drug discovery. And I mean, this kind of area where in active learning, maybe not in the most hyped area, but it's really good to see. Yeah, we're showing a lot of problems across different applications.
Yeah. I think active learning, to be honest, is fundamentally the right thing. Yeah, do the right thing, yeah, and then you figure out how to do it in the sense of how do you integrate it with robotics and how do you learn and do you explore, exploit, and so on. So, but honestly, active learning does intrinsically make sense. Yeah, I think there is something in there, but you have to see how and where to apply it. I mean, I get that. Yeah, but companies do that. Yeah, for example, in phenotypic screening, that's a typical thing. Yeah. But happy to chat more next week then, for example. Yeah.
Right. It does seem like active learning is more used, if I understood correctly, like in the early stages. Yeah.
We're gonna
We
have to go.
say
Sorry.
bye-bye to the office. It seems like it's more useful in early stages, like generating, what was it, hit leads and lead optimization.
Yeah, in generative, in general, yes. Afterwards, the problem becomes very high dimensional. Yeah, so you don't have a single endpoint that you measure and so on. Afterwards, the question, I mean, you always have a profile of a compound, 100 properties. Yeah, maybe 10, one in the beginning, then 10, and then 100 you need to take care of. And you cannot, the active learning in that high dimensional space is very, very tricky. Yeah, and you don't have enough data to really have predictive functions that guide you. Yeah, so in the beginning, if you have 1,000 data points, single endpoint, you can do it. Afterwards, it's too high dimensional and too data sparse, yeah, to really automate it. Yeah. Afterwards, you need a bit of more expert opinion to do that.
Okay. I think it's better than to leave it the rest of the questions for one-to-one.
Yeah, sure.
We'll get in touch about those meetings.
Yeah, sounds good. Okay,
No, this is
great. Thanks.
amazing. Very good overview of the whole kind of field of how machine learning is used in biology or drug discovery. That was great. Thank
Yeah,
you.
sounds good. Great. Thanks for the invitation. Yeah. Just let me know and then we see you next week.